The wall of the uterus contains three layers: the endometrium, the middle myometrium, and the outer perimetrium. The inner endometrium fluctuates in properties throughout the menstrual cycle. The menstrual cycle has two stages: the proliferative stage, which is under the control of estrogen, and the secretory stage, that is under the control of progesterone. As the endometrium goes from proliferative stage to secretory stage, the thickness of the endometrium increases. In proliferative stage, the glands, they are tall and straight. However, in secretory stage, the glands, they become tortuous. In proliferative stage, the number of spiral artery is less. In secretory stage, the number of spiral artery increases. Okay, let's look at the histology of both of these stages. So I said that endometrium can be found in proliferative phase and secretory phase. This is the endometrium. This much is the endometrium, and just below the endometrium, there's your muscular myometrium. The inner wall of the uterus, or the lining of the endometrium, is simple columnar epithelium. Endometrium has two layers: the functional endometrium and the basal endometrium. Implantation takes place in the functional endometrium. You can see that the endometrium is around 2 to 3 millimeter thick. I mean, you can't see, but this is the information. It's 2 to 3 millimeter thick in proliferative phase. The uterine glands, they are round and uniform. All right. So now let's turn this endometrium from proliferative phase to secretory phase. In secretory phase, you can see that the endometrium has now become much thicker. So it has become thicker than before. Now it is around 5 millimeter thick. And the glands, which were round and uniform in proliferative phase, has now become tortuous in secretory phase. Okay, so these are the glands in various stages. You can see that in proliferative phase, the gland is elongated, straight, and tubular. In secretory phase, the gland becomes dilated, tortuous, and saw-toothed. That is, the inner appearance looks like the tooth of a saw. And it also gives off watery secretion, which is the uterine fluid. Even the secretory stage is divided into two: the early secretory and the late secretory. Let's look at all these three stages on the basis of two properties. Number one, the glands, and number two, the stroma. In proliferative phase, as I said earlier, the glands are round and uniform. They are round and uniform. And the stroma, it is closely packed. In early secretory, the gland becomes tortuous. And if you look at the epithelium of the gland, or the cells of the gland, you can see that just below the nucleus, there is subnuclear vacuolation. Just below the nucleus, there's subnuclear vacuolation. Due to these subnuclear vacuolations, see, there's your cell, the cell lining the gland, the glandular cell. There's the nucleus, and just below the nucleus, there's subnuclear vacuolation. Due to this subnuclear vacuolation, the gland gives off a piano key appearance. The dark key, the black key, represents the nucleus, and the white below it represents the subnuclear vacuolation. They look like the keys of a piano. And then you have the late secretory, where the gland becomes even more tortuous. The secretion has been put into the cavity of the acini. And then the stroma, you can see that it is comparatively loose as due to edema. It is edematous. All right. So these are the slides that can be given to you in anatomy practical. You can see that there's your endometrium. It's thin, around 2 to 3 millimeter thin. And it contains glands that are round and uniform. That's why this endometrium is in proliferative phase. Next, the endometrium has become much thicker. And the glands now have become tortuous. the glands now have become dilated and it gives up sawtooth appearance as well. So this endometrium is obviously in secretory phase. One last revision. So the endometrium can be found in two different phases, the proliferative phase and the secretory phase. In proliferative phase, the thickness of the endometrium is 2 to 3 millimeter. In secretory phase, the thickness of the endometrium is 5 millimeter. Here's the proliferative phase, 2 to 3 millimeter. In proliferative phase, the uterine glands are round and uniform. Here's the secretory phase, it is 5 millimeter. In secretory phase, the uterine glands, they are tortuous. They are tortuous, they are dilated, and they give sawtooth appearance. The stroma, it is compact, while here the stroma, it is loose or edematous. Ovary has to be the easiest to identify. Ovary, we all know that it is lined by germinal epithelium, which is made up of simple cuboidal epithelium. Ovary, it contains cortex, and medulla. The medulla contains blood vessels and lymphatics. The cortex is where the ovarian cycle is going on, where you have your follicular stage and the luteal stage. So you can see most probably the Graafian follicles. So in the histology slide right here, you can see ovary with Graafian follicle right here. For us to understand the histology of fallopian tube, all we have to do is study it in terms of the layers, like we did in case of GIT. So what we are going to do is look at the various layers of fallopian tube from inside to outside. So the innermost layer is obviously the mucosa, and the mucosa is highly folded and branched. Due to the branching of the mucosal folding, the lumen is almost filled up. As a result, you have very little or scanty lumen. The lumen is almost filled up. The mucosa contains the epithelial mucosa, which is none other than the ciliated simple columnar epithelium. The cilia helps in the transportation of egg from the ovary till the ampulla for fertilization. And then it also helps in the transportation of fertilized egg or zygote from the fallopian tube into the uterus for implantation. Then just below the epithelial mucosa, there is lamina propria. Then you have muscularis propria. Muscularis propria contains your typical inner circular and outer longitudinal muscular layers. So you can see in this histology as well, the mucosa is folded and branched so much so that the lumen is almost filled up. The identifying feature of vagina is its lining epithelium, and that is non-keratinized stratified squamous epithelium. The mucosa also has lamina propria that does not contain any glands. So there are no glands. The glands are not present in vagina. Now, the thing is that non-keratinized stratified squamous epithelium is also present in esophagus, and the histology of esophagus kind of looks like that of vagina because it also contains your mucosa and the muscular layer. But then the most important identifying or differentiating feature is that the mucosa of vagina, I repeat, the mucosa of vagina contains glycogen. As a result, it is pale stained as compared to esophagus, which is darkly stained. So you can look at the histology itself. There's the non-keratinized stratified squamous epithelium, and the mucosa, you can see it is very pale stained. This is a line directly from Inder Versing. Mucosa of vagina is rich in glycogen, so it is pale stained, and that is how you can differentiate vagina from esophagus. Till now, we have studied about the endometrium of uterus and the vagina. The endometrium of uterus is lined by simple columnar epithelium. The vagina is lined by non-keratinized stratified squamous epithelium. In between uterus and the vagina lies the cervix. The cervix has two surfaces, the endocervix and the ectocervix. The endocervix and the ectocervix. The endocervix is continuous with the endometrium of uterus. So, the lining epithelium of endocervix is going to be the lining epithelium of endometrium, and that is simple columnar epithelium. The ectocervix is continuous with the vagina. So obviously, the lining epithelium of ectocervix is going to be the same as that of vagina, and that is non-keratinized stratified squamous epithelium. Again, endocervix continuous with endometrium, so same lining epithelium, that is simple columnar epithelium. Ectocervix continuous with vagina, so same lining epithelium or stratified squamous non-keratinized epithelium. That means that there has to be a part or a point or a junction where the simple columnar epithelium of the cervix changes to stratified non-keratinized squamous epithelium of the cervix. That is where the endocervix changes to ectocervix, and that is the most important identifying feature of cervix. If you simply look at ectocervix, it is obviously going to have the same epithelium as that of vagina, and that is non-keratinized stratified squamous epithelium. But if you look at the junction between ectocervix and endocervix, you can see this. See, this is ectocervix and then there's endocervix. I know that because at this junction there has been a switcheroo, that is the non-keratinized stratified squamous epithelium has switched to simple columnar epithelium, and that is how this ectocervix has now become the endocervix. From breast anatomy, by now you must have known that breast is a fibrofatty gland. That is, it has two parts. A stroma, which is fibrofatty, that means it contains fibers plus fat. And by fat, I mean, obviously, you can see adipocyte cells. And then it has a glandular system. A glandular system. What you see here is the diagram of the glandular system that is present in your breast. So the breast has lots of acini. These acini, they together form a bunch called a lobule. Multiple lobule, they give off terminal duct, and these terminal duct, they merge to form a single lactiferous duct. Again, so you have multiple acini forming a bunch called lobule. You have multiple lobules, and from each lobule arise terminal duct, and these terminal duct from various lobules fuse to form a single lactiferous duct. So in histology, again remember, you have two things. You have the stroma and you have your glandular system. The stroma contains fibers and fat or adipocytes, since the stroma is fibrofatty. So you have adipocytes and lots of fibers. Adipocyte and lots of fibers. As for glandular system, as I mentioned earlier, you have these alveoli that are arranged in a bunch, and this is called a lobule. And there are multiple lobules which give off terminal ducts that end up in lactiferous tubule. Now, in between each lobule, you can see septa. Which is also called interlobular connective tissue. So in a histology that can be provided to you in your exam, this is what you can see. So you can see these various lobules right here. These are your individual lobules. And each lobule, they contain lots and lots of acini. And these lobules, they are separated from one another with the help of fibrous septum. And that's how you can identify mammary gland.